Good morning, everyone, or whatever it is according to your circadian clock right now. It's a pleasure to be here today and to open this session on the P factors. I will first very briefly introduce the general concept of these P factors, and then we will take this phenomenon to the next level with our independent genomic components of the brain. See if I can also follow. So in general, what is generally meant by the P factor is referring to what different psychiatric diagnoses and symptoms have in common, all captured by a single general dimension. So originally, the general psychopathology factor, or what was coined the P factor, refers to the correlations, a covariance, or co-occurrence of symptoms and diagnoses in the population that is captured in a factor analysis by a single P factor. Now, at the brain level, something similar has been derived as well, that has been referred to as a neural P factor, which captures patterns of MRI associations that are shared across different mental health symptoms and diagnoses. And similarly, at the genomic level as well, something called what has been coined the genomic P factor has been derived, that captures a DNA variation or SNPs that are associated with transdiagnostic psychopathology measures and different diagnoses at the same time. So I wrote a paper about how we can possibly interpret, or how I think we can interpret, these different phenomena at these different levels of investigation, and what the implications of this are for, in my case, my field of neuroimaging genetics in the context of mental health. And some key conclusions of this paper were that the neural and genetic P factor reflect a mixture of many, many different processes that are also somewhat correlated with one another. And it is due to this correlation of these underlying processes that this global P factor arises. And these are many correlated processes that can range from various things, from the very molecular, so protein-protein interactions, neurotransmitter, things in the synaptic plasticity, what have you, everything at the cellular level, but also things in the environment that include lifestyle factors and sociodemographic variables, for instance. Because of this complexity of what these P factors represent, their utility really depends on what you want to aim for. So if you're aiming to explain a lot of variance and to have a very efficient prediction model with few predictors but that are uncorrelated and that explain a lot in your outcome, then this is a very good strategy to use. However, if on the other hand you're interested in understanding more about the underlying mechanisms that drive this covariance, then deriving such a single factor is not the most useful, and other multivariate methods will be needed. And I'm more interested in the second goal, and I'm taking this more from the bottom up, so starting from the genome-brain relationships first. So more concretely then about my work that I want to present here. What we have available now, due to the generosity and the hard work of the Oxford group, is a huge amount of correlations, a very rich data set describing the pairwise associations of pretty much all common variation in the DNA, with pretty much all commonly measured variation in the brain as measured using neuroimaging. The question that we want to answer, or that I want to answer now, is how does the variation in the genome influence individual variation in the brain? And from this raw GWAS output, from this bunch of associations, we cannot directly infer that. The reasons for this are that the neuroimaging traits are super polygenic and ultra pleiotropic. So with polygenicity, what we mean in genetics is that any particular trait, in our case neuroimaging trait, is driven or is mildly associated with thousands and thousands of SNPs in the DNA. And none of these associations will be super dominant. So it will be very difficult to pinpoint one particular gene or protein that is driving this association, hence complicated to interpret. And with pleiotropy, we mean that any particular SNP or variant in the DNA that is associated, that is relevant for the brain, is associated with thousands of neuroimaging measures and modalities. So here again, these associations are subtle and widespread throughout the brain, if you will, for any particular SNP. So this might be difficult to interpret, but the good news is that we can actually take advantage of this polygenicity and pleiotropy using multivariate analysis. So because the genome-wide associations are shared between the different neuroimaging traits, we can actually combine them, just like with the P factor, and derive a single general genomic brain factor. And there are various ways to do this, and these papers also use different methods to derive what I can call such a general genomic brain factor, capturing the overall shared genetic associations with brain structure and function. And this method explains variance, or this genomic brain factor explains variance effectively, just like with the P factor. It also increases signal to noise because it combines the signal across different GWASs, and this can help to make it more reproducible. However, if you want to interpret these underlying mechanisms, it is very hard still, because similar as with the P factors, this general factor is driven by a combination of lots and lots of different specific processes. So if you want to understand these intermediate specific processes better, I think we need to model them in a way that matches their multidimensional nature. So instead of deriving a single general factor, it can be useful to derive multiple specific factors that ideally also map to specific underlying mechanisms. Let's rephrase that a little bit. So we have observed associations that are the result of a mixture of underlying hidden processes. Now we want to recover these underlying hidden processes that are the sources of these observations. This might begin to sound somewhat familiar to people in neuroimaging, because what we're really looking for here is a source separation problem that is often also the problem with EEG or fMRI. And for this, in neuroimaging, we tend to use independent component analysis, or ICA, for instance. And so we try to apply ICA to this genomic brain imaging associations. And we adapted the melodic algorithm together with Christian Beckman, who developed the melodic algorithm to do this. And as input, we took the Z-transform, so the standardized GWAS betas from the Oxford group's GWAS results. And instead of the voxels that we normally have in fMRI, we now have 1 million SNPs in that dimension. And instead of time, or TR, we have 2,240 MRI traits. And so we decompose this matrix using ICA into independent components. And on the other side, we get, for every component, a vector of MRI trait weights and a vector of genome-wide SNP loadings. And the independence is maximized in the SNP dimension. So to make this more concrete, I will show you the example of one output of one component. We now have derived 16 independent genomic components. I will start showing the genomic dimension of this. And here you see one of the components. It's not the only one that is interesting, but it's component 2. And we see here what is typically shown in a Manhattan plot, what is typically used in genomics. And instead of absolute p-values, we here have relative p-values. So every SNP is represented by a dot, and the y-axis represents the relative significance of that SNP, of the loading of that SNP on this component compared to all the other SNP loadings. So it's like normalized. And this component is driven at a genomic level very much by a locus on chromosome 17, as you can see here. And that maps directly onto corticotropin-releasing hormone receptor, which is, of course, very important for our stress response. Another gene that is directly next to that, so we can't really disentangle these two genes here, is a gene producing tau protein, which is relevant for neurodegeneration. Then there are some other less strong signals, but also signals mapping to this one component, and two signals on completely different chromosomes that are not correlated within the genome ended up in this one component, and both encode myelin basic protein. Those are the green ones here. And another gene that I think is quite important is the psinin gene, which encodes a protein that directly interacts with tau protein. And so we see that there are different signals that are at different ends of the genome that are put in this one component based on the similarity of the patterns of the associations across neuroimaging traits. In the neuroimaging dimension, this component is mainly driven by the contrast of diffusion-weighted MRI, and mainly also global structural MRI measures, total surface area, total white matter, and so on. And then if you look at which of the more specific volumes that is not global is associated with this component, it was the ventral diencephalon volume, which is the hypothalamus, which is where corticotropin-releasing hormone is released. You can browse our components in a web page that has been developed by my postdoc, Nicolo Trevisan. It's still somewhat under construction. We're constantly adding more and more to it. I'll show a QR code at the end of the talk so you can browse there. We very much also welcome your input. If something looks very striking to you that we might not have noticed, because there's a lot of information there, we would welcome it if you just email us, or we can discuss it in more detail. If you want to read more about our method and what this is all based on, here is a paper that where we describe this in detail. We also show in this paper that this genomic ICA effectively reduces the dimensionality of the data. So 10 components in this paper explained about 40% of the variance of over 2,000 genome-wide association studies. So that is a reduction of 2,000 to 10, whilst maintaining a lot of the variance. We also show that it increased reproducibility compared to the original GWAS, and that this decomposition is largely driven by multi-modal MRI traits. So that indicates that the multimodality is needed to capture independent genetic signals. Now we are moving on, and this is more work in progress, to try and see to what extent these components also describe meaningful inter-individual variation, because so far we've just been looking at the summary statistics space. And eventually, we want to use this to also stratify etiological heterogeneity or other types of heterogeneity that are very relevant for mental health and potentially clinically relevant. But first, we want to just build this from the bottom up. So we want to see to what extent do our components describe inter-individual differences in the brain in the first place. So we took over 3,500 individuals who had not been included in the original GWAS, and we computed individual polygenic component scores for these individuals based on our independent components. And we did this in a way that is very, very similar to standard polygenic scoring. So we took for every individual, for every component, the individual SNP genotype, and that multiplied by the SNP loading for that component, and the sum of that will be that individual's component score. So with that linear combination, we end up with 16 polygenic component scores for every out-of-sample individual. The first observation is that these polygenic component scores are very uncorrelated. And this is useful because we want to describe inter-individual variability effectively. And if we have correlated variables, this is not a very effective way to describe inter-individual variability. It is also not exactly trivial, because the original neuroimaging traits are highly genetically correlated. If we had just simply derived polygenic scores from them, we would end up with very correlated variables and a not very effective way to describe individual variation. So we have effectively decorrelated genetic associations on different brain features. Then we also see that our genomic components explain quite a lot of variance in this out-of-sample individuals. And for the diffusion traits, as well as for the global brain metrics, we explain 6 to 12% of the variance, which is really a lot in terms of polygenic scores. Standard polygenic scores explaining maximum only 6% of the variance. For other traits, we explain less variance, and we think that this is because they might be more noisy, and likely larger discovery GWASs will allow us to further increase the explained variance there. So in conclusion, the neural and genetic so-called P factors are the end result of many biological processes that are relevant for brain and mental health, and they are all highly correlated with one another. When we developed genomic ICA, and we applied this to many neuroimaging traits, and we are finding strong indications that this can decorrelate and help disentangle ultimately these underlying processes, and we hope that ultimately this can help us to, for instance, identify meaningful variation also in clinical populations. I would like to thank my former PhD student, Serena Soheli-Nezat, who started this project with me and Christian Beckman about 5 or 6 years ago, and then my current PhD student, Leonard Oblong, and postdoc, Nicolas Trevisan, who are continuing this now. And here you can see on the QR code, you can browse through to the atlas, the component atlas, and feel free to contact us if you find anything there, and maybe there's a way to collaborate on interpreting these components. Thank you for listening. And,
thank you, Emma, for an excellent presentation. We decided that we'll take one question after each talk, and then we'll take more questions at the end. So if anyone has a pressing question right now, please go ahead.
Yeah, if I may, thank you very much for the great presentation. I was wondering about if you checked or looked at the polygenicity of the components. And where I'm going at is like, are they real endophenotype and simpler than the brain measurements? Or are they meta measurements and very complex genetically?
Yeah, that's a great question, thank you. The components that we see are mostly very polygenic, although some have, like I've shown, a single peak that really drives most of it. The ones that we have now also have other loci that also drive it. And there are some that I didn't show because it's harder to also interpret that are completely polygenic, just like other traits. I think that in the future, if we can extract more components, like say 100 or 200, which we initially tried, but it's not very reproducible at this point. But in the future, if we have bigger GWASs, we can do that, and then we will get to more, probably also single locus components as well. Yeah.